Hello students, this is your Prasanna ma'am from NEET World. Today I am going to discuss about RNA world and types of RNA. Learning this concept will give you a better understanding on transcription, translation and replication as well. This will give you a complete foundation on how proteins are forming from RNA, and this is to be focused to maximum extent both for NEET and EAP set. Okay, let's get started. From Hershey's and Chase experiment, it is unequivocally proved or finalized that DNA is the genetic material, not RNA or proteins. Okay, but then from RNA world, it is experimentally proved, and there is an experimental evidence that RNA is the first genetic material. What is the first genetic material, ma? RNA is the first genetic material. How it is said, like RNA is the first genetic material, not DNA, and it is also said that DNA has arisen from RNA. DNA has arisen from RNA through chemical modification. DNA has arisen from RNA through chemical modification of RNA. So if you look into it keenly, DNA is formed from RNA, they are saying, and RNA was the first genetic material. So at that time, all the essential life processes, all the functions of essential life processes are conducted by RNA only. All the essential life processes were conducted by RNA only. How and why? What type of activities are conducted by RNA? RNA can act as catalyst. RNA can act as catalyst. So they can help in catalysis. RNA conducts translation. And it can synthesize proteins. RNA plays major role in splicing. So all these life processes or essential roles are conducted by RNA, but in the current statement we say that DNA is that storing the information, genetic information, not RNA. Why is it so? Why DNA is storing the information, not RNA? We say that DNA is storing the genetic information, RNA is transferring the genetic information. Why is it so? Let us see. If you look over here, both the DNA and RNA structures. In DNA, what sugar you have, Pata? In DNA you have ribose sugar. In DNA you have deoxyribose sugar. In RNA you have ribose sugar. If you look into ribose at second carbon, this is C1, C2, C3, C4, C5. This is a pentose sugar, right? Both deoxyribose and ribose are what, ma? Pentose sugar. So if you look over here, in second carbon of RNA of ribose sugar, you have a hydroxyl group. This makes the RNA unstable and reactive. Unstable and what, ma? Reactive. If you look over deoxyribose structure, you don't find any such hydroxyl group. There is no such hydroxyl group. This adds stability to DNA. What does this do, ma? This adds stability to DNA. And second most important point is the nitrogen base. The nitrogen base, that is thymine, which is present in the DNA instead of uracil, is adding extra stability for DNA. The uracil which is present in the RNA is making RNA more reactive and unstable. So because of the free OH group that is present at the second carbon, it is more reactive and conducting multiple life processes. So what are the activities of RNA? What are the activities of RNA? We will learn in detail. If you see RNA, we have two types of RNA beta. What are they? Genetic RNA. We have two types of RNA, that is genetic RNA and non-genetic RNA. We have genetic RNA and non-genetic RNA. Non-genetic RNA. In genetic RNA, what does it mean? RNA is stored as genetic material, or RNA itself is the genetic material. You will see such type of genetic RNA in tobacco mosaic virus, QB bacteriophage, and retroviruses like HIV. In all these, RNA is the genetic material, and non-genetic RNA, which you will commonly found in mammalian cells, human cells, wherein RNA conducts many of the functions like catalysis, splicing and translation. Catalysis, splicing and translation. If you look over here, catalysis, how catalysis is being conducted? RNA which act as catalyst is called as ribozyme. RNA which acts as catalyst is called as what, ma? It is called as ribozyme. One example of that ribozyme is peptidyl transferase. This peptidyl transferase is highly useful, or this activity is to connect the amino acids at the time of protein chain formation. What does it do, beta? It connects the amino acids to form a peptide chain. Peptidyl transferase will connect, or it will bind two amino acids with peptide bond. It will bind two amino acids with peptide bond, and it will lead to the formation of a long protein chain. And if you see splicing is done by SNRPs and proteins. It is done by SNRPs and proteins, wherein SNRPs consists of RNA. If you look into RNA, heteronuclear RNA is formed at the end of transcription in eukaryotes. At the end of transcription, what is formed, beta? Heteronuclear RNA. This heteronuclear RNA has both introns and exons. It has both introns and exons. Okay. So always do remember, intron means inactive. It is rhyming, right? Intron means inactive. Exon means expressive. Intron means inactive. Exon means expressive. So these introns are spliced and exons are joined to form a mature RNA. What is happening? Introns are spliced and exons are joined to form a mature RNA. Then this RNA is utilized for the process of translation, and proteins are synthesized by copying what? mRNA, that is matured RNA. Okay. Next, how is it useful for translation? RNA used for translation by adapter RNA molecule. What is that? Adapter RNA or soluble RNA, that is tRNA. tRNA is called as adapter RNA because on one side it connects the amino acid, on the other side it codes for the protein. Okay, on one side it connects the amino acid, on the other side it codes for the protein. If you see, on one side it has connected the amino acid, on the other side it has coding for the protein. Okay, that is why it is called as what, ma? Adapter RNA. So what we have learnt over here? Genetic RNA and non-genetic RNA. Genetic RNA where RNA is the genetic material, for example in TMV, QB bacteriophage and retrovirus. If you look into the non-genetic RNA where RNA is doing multiple essential life processing functions like catalysis, it is done by ribozyme. One such example of ribozyme is peptidyl transferase. Next is splicing. Splicing is done by SNRPs and proteins. SNRPs include RNA, wherein it will splice the introns or inactive sequences from the heteronuclear RNA in eukaryotes. Okay. Once introns are spliced, then only exons will be present on the matured RNA that are copied for proteins. How come it is helpful in translation? By adapter RNA, that is tRNA. tRNA is called as adapter RNA because on one side it is connecting with amino acid, on the other side it is coding for the protein. That is why it is called as adapter RNA. It codes the codons which are present on the matured RNA. Now, we will learn what are the different types of RNA. What are excuse me, what are different types of RNA? Types of RNA. We have three types of RNA. Those are mRNA, that is messenger RNA. Second type is rRNA, that is ribosomal RNA, where we have already also learnt it in ribosomal RNA. Ribosome we have learnt it in Cell the Unit of Life also, know, where it has two subunits, larger and smaller, that included with RNA, that is called as rRNA. Third is tRNA, that is transfer RNA or adapter RNA or soluble RNA. rRNA is also called as insoluble RNA. It is also called as insoluble RNA. tRNA is also called as transfer RNA. It is also called as what ma? Transfer RNA. Okay, and this is also called as soluble RNA. Soluble RNA and adapter RNA. Adapter RNA. Now, let us learn each type of RNA in detail, which is very much important. Okay, mRNA or messenger RNA. Firstly, this accounts only 5% of total RNA. How much does this consist of? 5% of total RNA in a cell. And how many base pairs do it have? 75 to 6000 base pairs. What is the molecular weight of mRNA? 25,000 to 2 lakh Daltons. It carries a coding message for many amino acids. Of course, this message is being coded from double-stranded DNA. It is coded from what ma? Double-stranded DNA. This is coded from double-stranded DNA. Information of this strand is being coded to mRNA and the codons from here are further coded by tRNA. Okay. Next, this is short-lived. It is short-lived and commonly degrades after protein synthesis. mRNA, once it is synthesized at the end of transcription, once the proteins are being synthesized, the codons are copied by tRNA, the mRNA structure will be disabled or dismantled. It is also called as template, nuclear, messenger or information RNA. All the synonyms are very important. You have to keep it in your mind as it carries genetic information provided by DNA. Okay, all these synonyms are given because it is carrying the genetic information from DNA and helpful in the process of protein synthesis. This is all about messenger RNA, where you need to remember this, and this is most important. What polymerase is being used for the synthesis of messenger RNA? Once look at here beta. If you assume this as nucleus, we have already discussed nucleus has a non-membrane bound structure that is called as nucleolus. This nucleolus, inside nucleolus, what is synthesized is, inside nucleolus, rRNA is synthesized. Inside nucleolus what is synthesized beta? rRNA is synthesized. In nucleoplasm, tRNA and mRNA are synthesized in nucleoplasm. In nucleoplasm what types of RNA are synthesized? tRNA and mRNA. In nucleolus what is synthesized beta? mRNA. And what polymerases are there, that you have to keep it in your mind, and how much percent of RNA is present in each cell. These are utmost points to be focused. Next type of RNA is rRNA, that is ribosomal RNA. Here this accounts 80% of total RNA in a cell. How much? So the most abundant RNA is what ma? rRNA, and the least is mRNA, and it is degraded once the protein is synthesized. How many bases are present in rRNA? 100 to 500 bases are present, and the molecular weight of this is 35,000 to 1,80,000 Daltons. This is smaller to moderate in size. What is the size of rRNA? Smaller to moderate size. And the synthesis occurs in nucleolus. This point is utmost to be focused. And what RNA polymerase is there? RNA polymerase 1 is synthesizing rRNA, and it is most stable and used again and again. It does not degrade like mRNA. rRNA is more stable, it is not degrading like mRNA once the protein is synthesized. It is called as insoluble RNA because ribosome acts as structural component. Ribosome acts as what ma? Structural component. This is not soluble in 1M sodium chloride. This is not at all soluble, as this is acting as structural component. Larger subunit, smaller subunit, they are tightly bind, and inside the larger subunit and smaller subunit you have further subunit proteins. Okay, what are they? If it is larger subunit, that is 80S. If it is larger subunit, that is 80S, where the larger subunit is 60S and smaller subunit is 40S. Here what proteins are present? In smaller subunit 18S proteins are present in eukaryotes of 80S RNA, and in 60S, that is larger subunit, 28S, 5.8S, 50S rRNA are present. Just remember one thing, all 8s are there over here in eukaryotic 80S rRNA, wherein in larger subunit you have 28, 5.8, 5S rRNA. Here in smaller subunit you have 18S rRNA. Both in prokaryotic ribosome, that is 70S ribosome, and in eukaryotic ribosome, one protein subunit is common, that is 5S rRNA. This is common in both prokaryotic and eukaryotic ribosome. Okay, and along with 5S, here you have 23S rRNA. In smaller subunit it has 16S rRNA. Here it has in eukaryotic smaller subunit 18S rRNA. This is important, please do remember in your mind with these tricks. Okay, next, tRNA or transfer RNA. It accounts for about 15% of total RNA. How much? It accounts for about 15% of total RNA and in the cell. It consists of how many bases? 73 to 93 bases. The molecular weight is 25,000 Daltons. It is the smallest and coiled like a clover leaf model. If you look into the tRNA structure, it has two structures. One is secondary structure and tertiary structure. Secondary structure is called as clover leaf model. It is called as clover leaf model because it seems to be like that. No, it seems to be like a leaf, clover leaf model. If you look into the tertiary structure, this is called as inverted L shape. What is it? Inverted L shape. Clear? So if you look here in cloverleaf model, closer to the 3 prime end, amino acid is attached over the 3 prime end. Closer to this 3 prime end, there is a loop called as T-psi U loop, okay, T-psi U loop, and opposite to this there is an anticodon. This codes the information which is present in the mRNA. This codes the information which is present in the mRNA. This D arm is called, which is closer to the 5 prime of tRNA. This is called as D-psi U loop or amino acyl synthase loop. What is it called as beta? Amino acyl synthase loop. D arm called as amino acyl synthase loop, wherein it synthesizes the amino acids like this. Amino acid chain is formed like this, okay. For each amino acid, one specific tRNA is required. For each amino acid, one specific tRNA is required. One tRNA for one amino acid. One amino acid. For one amino acid, one tRNA is required. And for stop codons, no tRNA is required. tRNA is required for initiator and all other codons, but not for stop codons. Those are 3 codons, right? Next, what is this tertiary structure? That is inverted L shape. tRNA is inactive in tertiary structure. tRNA is inactive in tertiary structure. tRNA is active in secondary structure. Now if you see, it is synthesized by what polymerase? RNA polymerase 3. It is synthesized by RNA polymerase 3. RNA polymerase 2 is synthesizing mRNA in nucleoplasm. RNA polymerase 1 is synthesizing rRNA in nucleolus. RNA polymerase 3 is synthesizing tRNA again in nucleoplasm. Do remember this in your mind, okay. And about 5% of its bases are modified. How many bases? Only 5% of this bases are modified. This is also stable. So when it comes to stability in the 3 types of RNA, which is most unstable, you tell me beta? mRNA, because it is degraded once the protein chain is formed. Once the amino acids are forming, mRNA will be degraded. Next, and this also is used again and again. This degrades very slowly. This is called as soluble or adapter RNA. It is called as soluble RNA because it is easily soluble in 1M sodium chloride, and it is called as adapter RNA because on the one side it is coding the amino acids, on the other side it is coding for protons. Clear? These are all the 3 types of rRNA. Now let me give you a gist on how the protein is formed. What is the role of RNA in protein synthesis? Just give me a minute. Yes. So if you see from a double stranded DNA, all the codons over here and here. From a double stranded DNA, single stranded mRNA is formed copying one strand of DNA. Copying what ma? One strand of DNA, that is template strand of DNA. Okay, and this mRNA is similar to the coding strand of the DNA. The synthesis will always take place from 5 prime to 3 prime. Once the codons have been copied and coded to mRNA, at the initiator codon, let us assume AUG, at the initiator codon, this ribosomal subunit, smaller ribosomal subunit and larger ribosomal subunit will come and bind over this mRNA, okay. And this ribosome will have 3 sites over it, that is A, P, E site. What are they beta? A, P and E site. tRNA will come, first it will land on the A site, then it will move to the P site. It will move to what ma? It will move to P site. After coming to the P site, here it will copy the codons whichever are present over the mRNA, and from the amino acyl synthetase loop it will form the amino acid chain. Like that, for each amino acid one tRNA will form, it will move to the exit side and it will move on again. Then one more tRNA will come, land on A site, then on P site, ribosome will be moving on the mRNA, it will be coding for the next amino acid. So, the movement of the ribosome will go across the mRNA and the movement of the tRNA will go, and as such amino acid synthesis or amino acid chain is formed, right. This is the gist of transcription and this is the gist of translation which I have explained for protein synthesis. Tomorrow we will discuss in detail about replication, transcription and translation. Clear beta, from molecular basis of inheritance. As of today we have learnt what beta? RNA in RNA world, why RNA is the first genetic material, and currently why in DNA genetic material is stored, why RNA is unstable, and what are the different types of RNA, what are the most focused points of different types of RNA. This all we have learnt today. Thank you.